research. So we all know that the world is full of microorganisms, be it bacteria, virus, fungus, protozoa, and also I'm sure you all must be aware that our human body is more of the microbes. So you see, like I have given a picture where you can see that the human body is filled with microorganisms everywhere. So we are living in the world of microorganisms, we can say. Now, some of the microorganisms are beneficial to us, while others cause disease. So now, whatever are the beneficial microorganisms, we need not be concerned about that. But the microorganisms that cause disease are really concerning for the research world. And that is where the research comes into picture. So here you can see that I have put on some pictures of like diseases which are caused by bacteria. This is a tongue infection caused by Candida. This is also a fungal infection in dogs. This is a bacterial infection. And these are the fungal infections that we see in the plants. So no one is, like the microorganisms don't spare any living organisms from their infections. So now let us see what are antibiotics or what are antimicrobials. So this antimicrobials or antibiotics, they are chemical substance which is produced by one living organism. And generally that living organism is a microorganism. And then that chemical substance is detrimental to other microorganisms. Either it will inhibit the growth of the microorganism or it will kill the microorganism. So here I have put a picture where you see this is a bacteria, red one, and the green one is a bacteria. However, the red bacteria is producing some chemicals that is causing the lysis or the breakage of the cells of the green bacteria. So here you can see that this is a red bacteria which is producing certain chemical substances which you can call as antimicrobials or antibiotics. And then that is causing the lysis. So when these antibiotics come in contact with the cell wall, or it can also break the DNA or act on the RNA or proteins of the bacteria, thus causing cell lysis of the other bacteria. Now, what are the different types of antimicrobials? So they can be antibacterials, they can be antifungals, antivirals, and antiparasitic. So antibacterials are those drugs that are that acts against bacteria. That is like the bacterial pneumonia, drugs for bacterial pneumonia. Antifungals are the drugs for yeast infections, like any Candida infections or the ringworm. And then there are antivirals, which are the drugs for herpes or HIV. And then we have also the antiparasitic drugs, which are the drugs for malaria or any leishmaniasis or any parasitic that acts on any parasites. Now, what are the different source of antimicrobials? By microorganisms, mostly I'm saying. Mostly they're produced by microorganisms, and those chemical substances act on other microorganisms. So this is actinobacteria. This is a type of bacteria, and I have put in this picture because this photograph was taken by me during my PhD. And I have worked like, I think around 8 to 10 years, I have worked on actinobacteria, which is known as the reservoir of antibiotics. So another source of antimicrobial is fungus. Antimicrobials have also been reported from insects, from snakes, that is the snake venom, from frogs, from sponges which are present in the sea or the ocean. Antimicrobials have also been reported from plants like basil plant, neem plant, and there are other groups of bacteria apart from actinobacteria. So now, as I said, during my research career, I worked on the isolation of antimicrobials from bacteria, and mostly I focused on actinomycetes. So actinomycetes is a phylum of bacteria. So this is how the actinomycetes looks in the plate. So I'm sure like you all have seen bacteria in the plate, like, is it? Yeah, I'm just waiting for the response from the audience.
Yes.
Yes. So how does the bacteria look like in the plate? So mostly the bacteria will be like whatever other bacteria. So it will be like shiny, slimy. But this actinobacteria, it is dry. You can see that these colonies are dry, and they are known as the connecting link between fungus and bacteria, because they are filamentous in nature, just like fungus. So these are all actinobacteria. They are dry, they are chalky, powdery in nature, these colonies. So during my PhD and my postdoc, I have isolated this actinomycetes from Northeast India. So I'm from India and I come from the northeastern part of India, that is Assam. So my land is famous for tea and one-horn rhinoceros. So Northeast India is a part of the Indo-Burma mega biodiversity hotspot. So in total, there are 34 hotspots in the world, which is reported in this paper. And Indo-Burma all the red marked points are all hotspots. And Indo-Burma mega biodiversity hotspot is somewhere here. So this is where, like in biodiversity hotspots, you find a lot of flora, fauna, different types of microorganisms, different types of plants, and there is a possibility of finding novel compounds from these hotspots. So this is the Indo-Burma mega biodiversity hotspot. So, and this is the map of India. So Indo-Burma mega biodiversity hotspot is here, and it also includes Burma, Myanmar, and China. So from everything okay?
Hello.
Sorry.
Let's keep off our mics, please.
Okay. So I have collected. So this is the map of India.
Hello, Dr. Priyanka. Maybe you can keep off or mute.
Yeah. Okay.
Yeah, you can mute all.
I'm not able to mute others from I think because you are the host, so I think you will be able to do it. But anyways, yeah. So you see the blue part here. So this is Assam. So this is the map of Assam. And then from Assam, I have collected soil samples from here and also from here. Okay, this pink blue and the pink spot is where I have collected my samples from for the isolation of bacteria. And this I have mostly, my collection sites are national parks and wildlife sanctuaries from Assam or Northeast India. So why? Because the protected forests are not there is not much human activity there, just like any other place that we see. That is the reason, like, it is virgin, we can say. So I have collected samples from forest. Oh, yeah. So coming to the isolation of actinomycetes from the forest. So I have collected the soil sample from the Pobitora Wildlife Sanctuary, which is present in Assam. And I have isolated, like this is during my PhD, I have isolated 107 actinomycetes from this part. So again, during my postdoc also, I have taken I have collected samples from forest ecosystems of different states of Northeast India. So there I have collected around, like, I have done endophytes at that time, and this is soil. So there I have worked on around 200 actinomycetes. So yeah, so during my research career, I have worked a lot on this bacteria. So now, after I have collected the soil, and then now I plate it on a agar plate. So you see, with which is already embedded with some antibiotics, so that it reduces the growth of fungus or any other type of bacteria. Why? Because actinomycetes is slow growing. So you all these colonies appear only after 7 days. And then the colonies can also grow up to 28 days. After 28 days, also colonies can appear. So that is the reason we put some antibiotics so that it is easy for the actinomycetes to grow here. And then like the red colonies that I have marked here, so that is the actinomycetes. Now this actinomycetes, I have grown in a broth, that is a liquid culture. And then now my main aim is to isolate antimicrobials from the actinomycetes. So I have grown it in broth. I have done the ethyl acetate extraction. That is, I want to take out the antimicrobial compounds from the broth. And then here is the pure extract after the antimicrobial activity. This is the dried extract. And then I checked antimicrobial activity against, suppose this is Staphylococcus aureus, this is MRSA, E. coli, and Candida albicans. So this is by spot inoculation method. So all these colonies that you see, they are actinomycetes, different actinomycetes, and they are showing big zone of inhibition, means there is no growth of Staphylococcus aureus. So this is Staphylococcus aureus, but there is no growth of Staphylococcus aureus here. Why? Because this colony is producing antibiotic or antimicrobial substances that is inhibiting the growth of Staphylococcus aureus. So this is how I have done this spot inoculation method. And then I have done the disc diffusion method. So these are small discs where you load around like, how much can you load? Around like 40-50 microliter of your sample. That is the extract that I have shown in the previous slide. So you just put the extract and then let the organisms, let the test organism grow. And then here we can see that there is big zone of inhibition, means this extract is not allowing the MRSA to grow here. Now, I have also done some scanning electron microscopy to see how these antimicrobial substances have acted on the pathogens. So here, this is the MRSA, this is the control MRSA, and this is the control Candida albicans. So when I have, this is the control MRSA, this is the control Candida albicans. So when I have incubated these cells with, when I have incubated these cells with my extract for 24 hours, so you can see that there is change in the morphology. So there is breakage and shrinkage of the cells even here. So I can say that my compound, my extract has antimicrobial substances. And this is again Pseudomonas aeruginosa, and this is Candida albicans. So these are the control cells, that is without any antimicrobials. And this is after incubating with the antimicrobial for 24 hours. You can see that there is change in the cell morphology. And also you can see that here there is cell breakage. So this confirms that, the extract has antimicrobial activity. Okay. Now, if let us now suppose my topic here is to speak about proteomics and metabolomics. Now let us look what are the different omics tools. They are genomics, that is study of the entire genome of an organism. Genome is the DNA. Then there is transcriptomics, is the study of the entire RNA of an organism or a cell or a tissue. Proteomics, that is the study of the entire proteins of an organism. And metabolomics is the study of the entire metabolome, that is the different, the combination of all the metabolites which are produced by an organism or a cell. So these studies are omics. So I'll be concentrating on proteomics and metabolomics. So now coming to proteomics. So what is the proteomics? It is the study of the entire protein of a cell or a tissue or an organism. So the proteome, it can be of a species, that is Homo sapiens or E. coli, that is the entire protein of the E. coli culture, or it can be of an organ, that is the proteome of a liver cell, or a kidney cell, or any cancer cell, or cancer cells, or also it can be of a particular cell. And the proteome is not constant in the living body, so it differs from cell to cell. The proteome of the liver will be different from the proteome of the kidney, and it also changes from time to time. Like when a baby is born, his liver proteome will be very different from when that person reaches 60 years of age. So it changes over time. And here I have just tried to show a bacteria that is producing this small like proteins, and these are the protein structure. So this was also during my PhD, where I have worked on antimicrobial proteinaceous compound from Streptomyces KZ47. So this organism is already in the NCBI database. So here this is the spot inoculation method against Candida albicans. This is Candida albicans, and this actinomycetes, this Streptomyces is an actinomycetes. So this actinomycetes is producing antimicrobials, which is also confirmed in the well diffusion method, where in the well we load the extract. But now this extract I have not extracted using ethyl acetate, because this is a protein. So I have done the protein isolation, and then this is the SDS page that I have done. So here I have in the SDS page, I have checked the antimicrobial activity. Like I have cut the band, extracted the compound, and then I have done the antimicrobial activity, and then I checked that this is showing a clear zone of inhibition against Candida albicans. So I can so I came to know, like, this is the marker. So I came to know that this does not have any antimicrobial activity, but this has antimicrobial activity, and this is the entire protein of Streptomyces. And here I can see that the molecular mass is 14.3 kilodalton, so which is confirmed here. Now coming to metabolomics. So what is metabolomics? It is the study of the small molecules, which are known as metabolites, within the cells or the tissue or an organism. And these small molecules and their interaction within the biological system is known as the metabolome. So from protein we get the word proteomics. From metabolite, that is study of metabolite, is known as the metabolomics. So here this is an actinomycetes colony. Okay, this is a colony of actinomycetes, and you see these droplets. So these are the droplets that are produced by this particular actinomycetes. After, like, you can see these droplets after around 5 to 6 days, you can start seeing them. And these are the active compounds that act against any bacteria or fungus. Means it is antimicrobial in nature. So now coming to metabolomics, I have put this picture because I really liked it and I thought it will be easy demonstration. So now metabolomics, as I said, it differs from person to person, from cell to cell, from organism to organism. Now let us take a person from India and a person from Africa. Okay, like, a person from India and Africa, the climatic condition is different, the diet is different. Means these are the exposures, that is diet of both the persons are different. The medication they take will be different when they're diseased. Their physical activity will be different. Again, they are exposed to different pollution levels. So this is so all these things, all this exposure, it has an impact on the overall metabolomics. Means different metabolites will be produced by a person from India, which will be different from the person in Africa, because they are exposed to all these different conditions. And so there will be a metabolic response, and the disease risk will also be different. Because why? Because there will be changes in the level of some enzymes, or some different molecules will be produced, which will not be produced maybe by the person from Africa, and so he'll be like immune to all those things, but the person from India might be quite susceptible. So this is how metabolomics change from person to person, from cell to cell. So during my PhD and also my postdoc, I have worked on metabolomics, where this is the extract. I have taken the extract from actinomycetes. And then here I have done a GCMS study. I have done mass spectrometry study, that is GCMS, and where this compound, that is 3,5-base 1,1-dimethyl ethyl phenol, I have reported for the first time. And this paper is already published. So metabolomics, there are different tools or techniques or instruments to study this metabolomics and also proteomics. Now, what are those instruments? They are LCMS, that is liquid chromatography mass spectrometry. There is GCMS, gas chromatography mass spectrometry.
Yes. Sorry for the interruption. Just kindly, maybe you can make me a co-host so that I can mute those who are unmute.
Pardon?
Kindly, you can make me the co-host, please. You are currently the host.
Oh, how to do that?
You can go to my you can go to my profile and check the options there available.
Second, recording and caption, disable annotations. Okay, remote control, annotate participants. Right. Maybe I'll have to.
Or maybe you can click on my name.
Yeah. One second. Let me check once.
Okay.
This. I am not able to do that.
Oh, you can see that option of make me host.
I cannot see the screen where all the hosts are there. Oh yeah, one second. Yeah, where I can make you the host, right?
Or no, co-host, please.
Co-host, make host. I can see make host. I cannot see make co-host.
Okay, maybe you can continue. Yeah.
I have made you the host. Okay, I have made you the host now.
Okay, let me try. I think because I should make you the co-host so that you can continue sharing your screen.
Okay.
Sorry, I can't also see that option. I can't see the option.
Host disable participant screen sharing.
Let's continue. I can see the option also. Let's continue.
Oh,
You are now the host, but now,
okay.
yeah, I would like to request kindly everyone, let's get muted, please. Bismarck, Abubakar, please, let's mute ourselves. Sorry for that interruption, Dr.
Oh, it's
Priyanka.
okay. It's okay. No issues.
Okay, let's go on. Thank you.
Yeah. 1 second. Let me make it full screen. Yeah. Is it visible? Is my screen visible?
Yeah, sure. You can continue.
Yes.
Thank you.
Okay. So now there are different instruments by which you can study the metabolomics and the proteomics. So they are LCMS, GCMS. So what is the difference between LCMS and GCMS? So this is liquid chromatography mass spectrometry, where you can identify the non-volatile compounds in LCMS, and in GCMS you can study the volatile compounds. So some compounds, when the instrument is subjected to high temperature, that is above 100 degree, so these molecules, they ionize, and then you can get to know what are the compounds in LCMS. So the at high temperature, the molecules will not be volatile. So you can study the non-volatile compounds in LCMS. And here you have the nuclear magnetic resonance, that is the NMR. You also have the ESIMS, that is electron spray ionization mass spectrometry. And this is the MALDI-TOF, that is matrix assisted laser desorption ionization and time of flight. It's so difficult to remember this big name. Yeah. So now let us see what is artificial intelligence. So it's artificial intelligence is a boom nowadays, like everywhere you see that is artificial intelligence, and people, students, scientists, they are using it to like any extent that they can. So now what is artificial intelligence? So it refers to the simulation of human intelligence in machines that are programmed to perform tasks and make predictions. That is, see, humans can make predictions, human can make recommendations, or humans can make decisions. But now, using some algorithms and some programs, the machines are made to do these things that are done by humans. That is, to make prediction, recommendation, or decision that requires human-like cognitive abilities that influence the real world or the virtual world. So what are these cognitive abilities? So cognitive abilities are thinking, reasoning, remembering, imagining, learning new words, and using a language. So these are the cognitive abilities which are human-like, and they have been put in using some algorithms and some programs. They have now been installed in machines to do work for us. So this is a brief history of the artificial intelligence in healthcare.
Hello, Dr. Priyanka. Yeah, sorry for that interruption. Yeah, your presentation, your slide is still in the previous presentation. So I don't know if I'm the only one observing
Is it changing?
That. It is still not changing.
Not changing?
Yeah.
Okay, so I'll just exit the full screen. Okay. You can see my screen, right?
Yes, sure. You can now see. You can put in this presentation mode and proceed. Thank you.
Okay. So, yeah, I think I will not do in the full screen mode. I'll just use like that. Yeah. So this is the history of artificial intelligence in healthcare. So I'll not go like in too much details into it. But as of now, like you see, the whole concept of artificial intelligence, it started in 1950. Okay. And then all this happened. And now we are somewhere here. That is AI trials in gastroenterology. So this has taken place already in 2020. And this is the paper which from where I have taken this. Then these are the classification of AI, like they can do perception. So first of all, reasoning and problem solving. Then knowledge representation, planning and social intelligence. So all these things, humans are making a machine to do that. Perception, machine learning, natural language processing. So these are the classifications of AI. So everywhere you will see that I have put the source of the paper from where I have taken this. Now, if we look at the use of AI, that is artificial intelligence in drug discovery research. So first, artificial intelligence is used in the research and discovery of different metabolites or different proteins that has antimicrobial activity. It is also used in clinical development. Clinical development is when a metabolite or when a drug undergoes the clinical trial, and then you have to decide whether the drug is good or bad. So that is like a machine is able to interpret that. So that is artificial intelligence. So that is clinical development. Patient surveillance. After you give the drug, how is the patient responding to the drug? So that is the patient surveillance, and also post-market surveillance. So after the drug is released in the market, how is the drug doing in the market? Like, is it getting sold, or, like no one is using it? So that is the post-market surveillance. Now, coming to the use of AI in antimicrobial research, they are used in clinical care, drug development, health system management, identification of new AMR. AMR is antimicrobial resistance, surveillance, and prediction-based diagnostics. Now, if we look at the AI-based proteomics, so the topic that was given to me today was AI-based proteomics and metabolomics. So now let us see what is the AI-based proteomics for antimicrobial research and discovery. Means how AI-based proteomics is helping in the antimicrobial research and discovery. First of all, target identification and validation. So AI can prioritize. So this is the source of the paper. So AI can predict like protein-protein interaction, then the function of a protein, and also it can prioritize the potential targets of antimicrobial compounds. So on what targets will the antimicrobial compounds act? Okay, and that is based on the expression pattern, protein-protein interaction, and functional annotation. So here you can see that this is the target discovery, means you discover a target, means that a compound or an antimicrobial compound, it will, suppose you have made an antimicrobial compound that acts in the liver, okay? It acts in the liver, and then in the liver, in which part, or it will bind to which amino acid. That is the protein-protein interaction. What is the active site? So that is the target discovery. First, you do that, and then you identify the hit identification. So what are the different hits that you get? Then the lead identification, lead optimization. So you optimize the lead, and then that particular lead, that is that particular compound, antimicrobial compound, goes for clinical development. So all these things can be also done by AI. You do the research, you do the wet lab experiments, but wet lab experiments take time. But artificial intelligence, it won't take much time. So later you can compare the two results and see like if your results match or not. So that is the target identification and validation. Next, you have the analyze. It is useful for analyzing the admitox. So if you all go for when clinical trials are done, so it is very important for the drug to undergo this admitox. So what is admitox? That is absorption of the drug. A is absorption. D is distribution of the drug. Distribution of the drug to different, suppose I have taken a drug. Now I scientists want to see how that drug has been distributed in my bloodstream, how it goes to the liver, how it is going to the kidney. And M is metabolism, means how the drug is getting metabolized in each of this thing, in each part of my body. Like, is it causing any harm to my kidney or something? Then you have the E. E is the excretion, how the drug is excreted from the body. Then tox is the toxicity study, whether it has any toxicity, toxic effect in my liver or my kidney. And then you study the physiochemical properties. So all these things can be done using the AI. So here it is, like compare it with database, then there is curation. Then there is MLDL, that is machine learning or deep learning, which is the artificial intelligence. And then you also do these experiments in vivo, and also you can do it by artificial intelligence. Next, coming to protein identification and function prediction. So these are the different uses of proteomics, AI-based proteomics. So AI can analyze the mass spectrometric data. So mass spectrometric data you get from the LCMS, from the GCMS, from NMR, MALDI-TOF. So those instruments, they give the MS data. And then, sorry. So this is the, so you get the MS data. And from there, you can identify the different proteins that is antimicrobial in nature. So here what you do is, like, this is, suppose you extract the protein, okay? you extract the protein, and then you get these candidates. So this is using the artificial intelligence, and then artificial intelligence using some algorithm, it will say that, oh, these compounds have antimicrobial activity and the ones with the cross do not have antimicrobial activity. Okay. And then they just do some molecular dynamics simulations to see like how the compounds act on different cells or the targets. So this one, you can also do the wet lab experiment, where now these compounds, you just do the antimicrobial activity and you see that, yes, artificial intelligence has predicted it right, that this compound has antimicrobial activity and this compound does not have. And this compound is a protein. So you can also do the protein identification and function prediction. Function prediction is whether a protein will be antimicrobial or not. So here we are concerned only about the antimicrobial activity. Now, coming to resistance mechanism identification, AI can also predict resistance mechanisms. So how? So AI can identify the expressed or the post-translationally modified proteins, which are associated with antimicrobial resistance. So what happens is, like there is a drug sensitive strain and there is a drug resistant strain. Okay. And then you do the you do the sequence analysis of that particular gene that you are interested in, and then compare the two genes, and then you will see that there are certain changes. There are changes in the nucleotide, and those changes are making a strain drug resistant. So you can identify that also by AI. Okay, so AI has made the life a little bit easier, but obviously validation should also be done. I would suggest that there should also be validation by wet lab experiments. AI can also detect protein-protein interaction. So it can predict the protein-protein interaction, that is interaction between the antimicrobial proteins and also the microbial targets. So and how this antimicrobial protein is acting against an organism which you don't want or which you want to kill it. So how that microbial target and how this antimicrobial protein is interacting, it is also, AI can also predict that. So here, this is a sequence-based structure modeling. So you already have the sequence, and from the new protein sequence, you can do a modeling. So suppose you have modeled this structure. This is a protein. That is the interaction. And now what you do is you match with the databases that are already available. Then you do the filter. You have to filter which is the potent, which you think is the potent. So you are not doing it. It is done by the AI. And here you are predicting the host microbe interaction. This is like the docking study. And then there is the network analysis, like using deep learning and machine learning. We are able to annotate that, okay, this protein, this is how the two proteins are interacting, that is protein from the drug and the protein from the microbe. Now, so you have seen that there are many, many different functions, but I have picked up only these important functions. Now, coming to the AI-based metabolomics. All this while, we have seen how AI-based proteomics is used for antimicrobial research. Now, let us see how AI-based metabolomics are used for antimicrobial research and discovery. So, these metabolites can be alkaloids, it can be terpenoids, caffeine, or any other small molecules. So, AI is able to predict antimicrobial activity. So AI can predict the antimicrobial potential of new compounds based on their metabolic profile. And this enables the identification of potent antimicrobial leads from large compound libraries. So you see, this is the, so all these are artificial intelligence simulations, which even I can't understand much because these are all like different algorithms, but I can understand the steps. So this is the structural data of the metabolite. Just let me have a little bit water, sorry. Yeah. And then using the data or using the compound, you can predict whether this compound will have antimicrobial activity or not. So this is the binary reading, that is 0101. So AI, they do some binary reading. They have some algorithms, and from there they see that this antimicrobial compound is acting against, or is acting against which different compounds. And then you can also predict whether the compound is antimicrobial or not, or how much antimicrobial, or whether it is showing any synergistic activity or antagonistic activity. So those predictions can be done using AI. Also, it is useful for metabolic pathway analysis. That is, AI can identify the metabolic pathways and the enzymes targeted by the antimicrobials. So the antimicrobials will act on certain enzymes in the test organism or in the pathogen, because of which the pathogen, so the antimicrobial will bind to the enzyme, and then the particular function will be stopped in the test organism or the pathogen, because of which the pathogen will die, or it will be a static thing, that is, the growth will be inhibited. And also it is used for understanding the antimicrobial mechanisms and also the resistance mechanism. So you see, like we do the sample preparation, then there is the data acquisition, data processing, multivariate analysis, and then there is the pathway analysis. So these are the steps how AI does the pathway analysis, that is where the particular antimicrobial activity is acting, which enzyme, whether it is an enzyme for DNA replication or a protein translation. So, and then, so this is the instrument. This is the, here they have shown an NMR here. Next, it can also predict this synergy and combination therapy. That is, synergistic activity, if two drugs act together, whether they have a synergistic activity, that is, whether they are increasing the growth of the pathogen, or whether they are completely inhibiting the growth, or whether they are acting together, whether they are killing the microorganism, or how is it acting. So even the synergistic and combination therapy is predicted by AI. And also this approach is helpful to overcome resistance. So when bacteria is exposed to too much antibiotics, so they develop resistance. So also this particular AI can also predict and try to overcome the resistance. It can reduce the toxicity, means it can predict, it cannot reduce by itself, it can predict the reduction of toxicity, and also help in improve treatment and efficacy. So these are like, in every step there is AI. Okay. And this picture I have taken from this paper. And then like suppose this is the drug, this is the drug combination, then you see the dose response. Also there is the drug combination. How is this drug acting with this drug in combination? So this is what happens, that is drug delivery system improvement. So there is good improvement. And then this drug is taken by the human. So here you do the ADME study, absorption, distribution, metabolism, and excretion. And here the outcome is that the prediction of the treatment outcome. So, for one person, that drug might be not a good one. He might show some toxicity effect, but for this person is showing very good effect. That can be predicted. Also, AI can also predict for metabolite identification, for that is identification of new metabolites. And this is done by integrating the metabolomics data, that is the mass spec data, with the databases and the spectral libraries. So spectral library is one spectral library is the NIST library, NIST library, which is used for the GCMS analysis. So you see, this is the biological sample. It can be a blood or urine. Then you do the mass spec, that is GCMS, LCMS, NMR. And then now the machine is trained, okay? Trained. From here, you do the prediction of how this metabolite, what can these metabolites be. Then This is the new, and then this is the in vivo study. That is when the drug, sorry, when the, this is the in vivo study, which is, that is this person's urine sample or this blood sample. And then you see that whether this metabolite is acting on the liver or the lung, how is it acting? Or how is his lung functioning, and how, what is the difference in function? So this is a total population of human being, and then you can see that this person is acting differently or his metabolites are different based on the artificial intelligence. You can do that. Sorry. Yeah, then also there is antimicrobial resistance prediction. So by comparing the metabolite profile of a drug resistant strain and a drug sensitive strain, so you can also see what are the, suppose this is a bacteria, you do the sequence analysis. This is the AI based prediction, then bioinformatics study. So you see that there is the sequence of the novel resistance gene, and this is the sequence of the sensitive strain, and this is the resistant gene strain. And then you compare this to, and then this is the, you can say that there is antimicrobial resistance in that gene. And then once you can identify that there is antimicrobial resistance, so you can give the proper medicine. So it will not be a generic medicine, it will be a personalized medicine. Personalized medicine only for that person. Next, also we can do, the AI can also help in predicting drug repurposing. So what is drug repurposing? Drug repurposing is that one particular drug, if it is already in the market, and suppose it is, that drug is used for liver function, like, you know, to enhance the liver function. So repurposing means we will study that drug also for other actions. Like we want to see whether the drug is acting against Candida albicans, or whether it is able to kill bacteria, whether it is able to, whether it has any anti-cancer activity. So that is known as drug repurposing. So by correlating the metabolite profile and the of the microbial pathogen with those of the known drugs, AI can identify drugs that exhibit antimicrobial activity against some specific pathogens. So this is the host cell. So these are the targets that like are predicted. And then AI will help you in saying that this drug can have what all activities. So you can see that there are different, like, you know, AI has been a boom, as I said, like it is used in metabolomics, proteomics, also genomics, transcriptomics, which I have not spoken in this lecture. So now let us see what are the future directions of AI in antimicrobial research. That is how AI is going to further help in the antimicrobial research. So that is integrating different omics data, that is integrating genomics, transcriptomics, metabolomics, proteomics, and what different omics are there. So integrating all the omics data will enable in understanding of antimicrobial mechanisms, resistance mechanisms, and also drug responses, means how a drug will act on a body. Then AI will continue to facilitate high throughput antimicrobial screening, means time. You can like deal with 1,000 you can do 1,000 antimicrobial screening just in a span of 5 minutes. And then of large compound libraries and virtual assays, because obviously it cannot do any in vitro, so it is a virtual assay. Also, it will contribute to the personalized medicine. So when you know the protein-protein interaction or how the drug is acting on the target, you can develop personalized medicine. Personalized medicine is for one particular person. It is not a generalized medicine for like gastroenteritis or something. So, you know, it will be a personalized medicine. And also it will aid in the translation of antimicrobial discovery into clinically viable therapeutics, development of novel antimicrobial agents. Some diagnostic techniques can also, like diagnostic techniques are also coming up where you can know that whether, you know, the person is infected with gram-negative bacteria or gram-positive bacteria or fungus. And also there is the precision treatment approaches, means already AI will predict like what particular medicine should be given, or like what is the exact problem that person is having. So that is the precision treatment will be available for that particular person. So yeah, thank you. Thank you. So if you have any question.
Oh, thank you. Thank you so much, Dr. Priyanka. That has been a very wonderful presentation. And
I
as far
hope
as
I
the
have done
use of
justice. I hope I have done justice to AI, because I'm just wondering that I can use AI soon in my research as well.
Sure, sure. Yeah, that was a very interesting and very intriguing lesson. And I hope that we have learned a lot, especially new techniques in, or maybe rather approaches in antimicrobial research and development, especially with this development of AI and informatics technologies, which is really revolutionizing healthcare. And yeah, I think this will be the future of
Yeah,
antimicrobial resistance, or rather healthcare.
yeah.
Yes, so I think, yeah, this is the Q&A session. So I welcome any question from anyone. Kindly raise up your hand, or you can rather post it on the chat box.
Yes,
Yes,
please.
for the next few minutes, please. So, Dr. Priyanka, as we wait for question from the audience, I'd like
Yeah.
to ask, one question, or how can we
Oh,
identify that?
someone has raised their hand, I think.
Okay,
Osmos. Yeah. Yeah, please carry on.
okay, so how can we identify a protein, like maybe, for example, if it is antimicrobial, like in silico, without going to the in vitro techniques? Can we identify that, or can we rule out that this particular protein has antimicrobial property purely in silico without going to the in vitro techniques? Thank you.
Yes, you can do like, but obviously it is also important to validate your study by doing in vitro studies. But yes, you can do like using the bioinformatics tools, where suppose you know the target. Suppose I am working on malaria, and I know that I want to study the effect of a drug against malaria, some protein, or take an enzyme for a particular enzyme, I'm saying enzyme A, and then I want to see the what is the active sites, at where all the active sites of that particular protein will be. And then based on that active sites, you can design different lead compounds. Suppose you design 1,000 lead compounds, and then you do the molecular docking, that is the simulation, and then you see like how that particular lead is acting on the active site. So you know most of the compounds will be like bound here, there, but you are not interested in that. So you see that there will be only a few compounds out of those 1,000. There will be only like around 20-30 compounds will go and bind to your active site. So then you can say, and also some bindings will be like hydrophobic, some bindings will be hydrophilic, some bindings will be very loose bindings. So you don't want that, because if it is a loose binding, it will not have much efficacy. So you want only those compounds which will be bound to your active site very tightly. And then once you get that, so from there, you can proceed. And then you can also make those molecules. You can tell Sigma or anyone to make those compounds for you. And once you know that, so you can also do the in vitro study to see whether that particular compound, whatever the bioinformatics or AI has simulated, whether it is really active on the in vitro study. So yes, that is possible.
Okay, thank you so much, Priyanka. Okay, that's well answered. Okay, we have Cosmos. Cosmos, kindly go on. Unmute and go on, please. Cosmos, we can't hear you well. or maybe you can type on the chat box, but rather you can also go on.
Yeah, like if anyone has, there is, yeah, I can see the chat box. Let me see. If anyone, yeah, it's if anyone has any question, you can also, yeah, do the like. Oh, sorry, I have. There is no question in the chat box.
Cosmos, can you hear me? Please, you can go on. You can write on the chat box. You can raise up your hand. Okay, it seems that maybe Cosmos is not getting as well. But meanwhile, also like to ask one more question is about on about heat identification of the proteomes or maybe proteins or metabolites. How do we identify these heats following in silico high throughput screening?
You mean the heat identification?
Yes, sure.
Oh, that's what. Like, so the heat, you can identify or you can design the heat based on your active site. So you already know, like, when you do a study, you already have do some background research on it. So you already know, like, you want, suppose you want, as I said, like, you want to see the act, you want to develop a drug against malaria. So now your next step will be which enzyme or which protein you will be targeting for Plasmodium falciparum, because malaria is caused by Plasmodium falciparum, Plasmodium vivax. So you do that. So based on that target, you will see the structure of the target. So based on the target, you will identify some active sites, and this active sites will where you will have the amino acids, different amino acids, because it's a protein. And then from there, you need to identify what can be the active sites, or what are the good active sites where the uh, molecule will be binding tightly to the this thing, tightly to the compound. Sorry, tightly to the protein. So, so there are different algorithms, and also, like, that is mainly the molecular docking, where that molecular docking comes into picture. So you can do that using Autodock, or so you can simulate that, and then Autodock will give you a clear idea, like how that molecule is interacting. And definitely you can also, like, do the in vitro study, like, by making those molecules.
Okay, thank you so much, Priyanka. And yeah, we have been getting a lot of students interested in use of AI and bioinformatics for antimicrobial drugs research and development, especially within our group, that is the Student Secondary Superbugs Africa. And yeah, definitely we're going to get back to you in case we get more questions or more research topics on the same. Yeah, we keep in touch. Yeah, because we have
so maybe
good.
you can share my email ID, like, if anyone has any question, so they can always get back to me, and I'm, like, quite active in LinkedIn as well.
Okay, sure. Thank you so much, Priyanka. I think, yeah, before we wind up the session, maybe I just would like to know briefly what is the uptake of AI and bioinformatics as far as antimicrobial drugs research and development is concerned within the UK, where you are at the moment?
So here, like, within my lab, we have not been using AI till now. So we have not used AI, but yeah, maybe in future we will be using. But definitely, like, bioinformatics is an integral part of the drug discovery labs everywhere. So there is a lot of use of bioinformatics everywhere, but we are yet to, do the whole thing or accept the artificial intelligence thing. So because artificial intelligence, like, it involves the work of the computer scientists, the mathematicians. So I think, yeah, like, since this is a very new thing, artificial intelligence, I think we will be using that soon. But yeah, we use bioinformatics a lot for our research, like doing any simulation study or maybe prediction of like different genes or analysis of genes, or if we do a whole genome sequence, if we have a whole genome sequence of an organism, we can also predict like what metabolites or what proteins are produced by those genes, whether those proteins or metabolites are novel or not. So those things we are that we are using bioinformatics. But maybe in future, yes, we will be using artificial intelligence as well.
Yeah, okay, yeah, sure. Thank you so much. I think, yeah, that is well and understood. Yeah, I can see Cosmos is back. Cosmos, if you can hear us, you can go on, please. Unmute and go on. Cosmos, over to you.
So sorry. Yes, it was a network issue. And my question was simple. I was like, can the migration from one country or a continent affect the metabolite of what do you call it? What doctor was presenting also? That was my first question. And the second one is there's misconception about getting infected when working with this bacteria via family research and stuff. So we would like doctor to also clear it for us. If it is true, what are the chances of me getting safety exposed? What are the clear all these misconceptions? Thank you so much, sir.
Hi, Jimmy. Can you please tell me the question, because I'm not able to follow? Sorry.
Okay, yeah, I think I did not get well the second question, but the first question he was asking about change of location. How does location affect the change in metabolites? Like, for example, in human beings or even the microbes. Like, say, for example, you're targeting isolation of some proteins or metabolites in a pathogen in different locations. How does that traveling affect the concentration as far as yeah, maybe amount of proteins and metabolites in the host?
Yeah, so that I have already said. So I'm saying it again. Suppose there is one person from Africa. Suppose we are taking Jimmy for example. Suppose Jimmy is a person from Africa and I am a person from India. Now Jimmy is staying in Africa, I am staying in India. So Jimmy and me, we both will be eating, we both are having different kinds of food habits. Our activity will be different. Okay. And then we are exposed to different environmental pollution. So whatever pollution or whatever pollutions Jimmy is exposed to, I am exposed to a completely different set of pollution. And then like food habits are different. Even if I am eating rice and Jimmy is also a rice eater, so the way we cook it are different. The place where the rice is grown in Africa, in India will be different. So all those things are contributing to metabolic changes in Jimmy and in me. Now, when you do a metabolic or metabolomic study, suppose you want to do a metabolomic study of my liver and Jimmy's liver. So you will see that because we are exposed to different diet, pollution levels, even our day-to-day activity, so, you will see that the metabolites of Jimmy's liver and my liver are completely different. Okay. So whatever I have will be different what Jimmy has. Even if there are two persons from India, maybe even within my family, I might see different metabolites for me and my brother or me and my mother. So like so many factors, they contribute to the change in the metabolites in the human system. And same thing goes for a plant also. A plant grown in Africa, that is a rice plant grown in Africa, will be very different from a rice plant grown in India, because they'll be exposed to different kind of pesticides. The soil will be different, the water, everything will be like different, and everything contributes to the metabolites. So I think I have answered the first question, and if anyone can tell me the second question.
Okay, yeah, thank you so much, Priyanka. I think another thing here, because we can see that disparity in location also contribute to the structural changes in the organism.
Yes.
So the variation in the structural makeup of an organism. So maybe, for example, that can also be attributable to the differences in the drug receptors from one organism to another. You can see that some
Right.
people are maybe or maybe susceptible to certain diseases, others are not, or maybe some
Yes.
drugs are
Yes.
effective in some individuals and in others it differs. So
And
that
also, like, even we can see that even in the family, even in the family, we can see that suppose it's a family of 5 members. Out of that, 1 or 2 members will be allergic to peanuts or will be allergic to sulfur
True.
drugs. But they are taking the same type of food and they are living they are exposed to almost the same kind of pollution and everything, all the environmental factors. But still, only 2 persons are developing that particular allergy. So there is a change in the gene, or there is a change in that is a single nucleotide polymorphism. It means there might be a change in one nucleotide which is contributing to that particular allergy. So like, so many different kinds of factors it contributes. So when I was during my PhD, I collected I worked on actinomycetes from 2 wildlife sanctuaries. Those wildlife sanctuaries are located in Assam, but in different parts of Assam. So Assam is a state within India. So within I have seen that even in when I have identified, like when I have isolated actinomycetes, so the actinomycetes from one national park is very different from the other national park. Also, the metabolites produced by one actinomycetes is very different from the other actinomycetes. So there is always changes based on the environmental factors. So
Yeah,
there
sure.
is one question. Yeah, there is one question in the chat by Jennifer. She has asked, why is personalized prescription required during antimicrobial resistance prediction with AI and not others? Why is personalized prescription required during antimicrobial resistance prediction with AI? I don't understand with AI and not others. So definitely, like, personalized prescription is required by personalized prescription is required because your body makeup is different from my body makeup. So suppose I am infected by Staphylococcus aureus, and then whatever drug is, like, made maybe for the generic population might not act on me. Why? Because I might already be resistant to Staphylococcus aureus. So whatever drug you are giving me, I am taking it, but then, like, I have not been cured. Why? Because there, like, already the Staphylococcus aureus is resistant in my body. So definitely you need a personalized medicine for me, that is only for me, which will act on that particular organism. Or like, as I said, like, there will be different metabolites produced in me and like a different person. So when you do give a personalized medicine, so this personalized medicine will be very much targeted, and then it is a precise medicine. So precise medicine only for me, because of which I don't need to keep on taking antibiotic, because taking more antibiotic is causing the antimicrobial resistance. I don't need to take more antibiotics. So because that is designed only for me, I take that and I'm cured. I don't have to take hundreds of antibiotics. So I can take only one and get cured. So that is that is one reason.
Yes, perfect. Thank you so much, Priyanka. We have now that question from Cosmos reads, misconception of getting infected when working on bacteria and most pathogenic microorganisms and the chances of getting infected.
Can you say a little bit slowly?
So it's about misconception. So he's asking about the misconception of getting infected when working on bacteria and most pathogenic microorganisms and the chances of getting infected. So it seems not very clear. So on my end, Cosmos, currently, maybe you can come up again very clearly because I still don't understand your question very well. But
or maybe Cosmos, you can always email to me. Yeah, you can always email to me.
Okay,
So my email ID is priya.sharma03@yahoo.com. So you can always ask Jimmy. So he has my email ID, and then you can always write to me.
okay, so of Cosmos, Can you hear us? Maybe you can come up again clearly, please.
Yes, please. I can hear, but my question
Cosmos, you are not very audible.
was if doctor can clear the misconception about bacteria, virus. Oh, so sorry. Can you hear me now?
Yes, very
Hear me
clearly now. It's okay now. Go on, please.
Yes, yes, thank you so much. I was asking about if doctor can help us to clear the bacteria and other microorganisms and the chances of getting infected. And because of that, most people try to dive their interest into research when it comes to bacteria and stuff. So if doctor who has already taken that kind of path can give us as to how we can go about them and clear the misconception that's not working with bacteria and virus and all the pathogenic microorganisms, you have a chance of getting infected. But instead, these are the other ways. If you don't do them well, there are high probability of you getting infected. So this is what I want doctor to clarify for us.
Like I'll just tell the question and then you tell me if I am like correct in understanding your question. So do you mean like, because there is like a misconception that if we work with microorganisms, like I work on microorganisms daily and I have been working on microorganisms, I think since 2009 or 10. So are you asking like if we work on microorganisms, we are more prone to getting infected? And if we don't work on microorganisms, so we are not educated about the microorganisms and then we have the chances of being infected more. Is it something like that?
Yes, please. That's
So yeah, the thing is like when we work on microorganisms, we take precautions. Like I work on microorganisms daily. So I take precautions daily. Like, when we work with, because there are these biosafety labs, that is CL1, CL2, CL3, CL4. So when you work with general microorganisms, which will not cause you any disease, so that you work in biosafety level 1, that is biosafety level 1. And then now suppose you are working with infectious organisms that can cause disease, that is like, but not very serious disease, but those disease can be treated, like you are working on Candida albicans or Staphylococcus aureus, or you are working on Plasmodium falciparum, so that you work on biosafety level 2. Now next is biosafety level 3. So currently in the UK, I'm working in biosafety level 3 in my lab, because we handle microorganisms also from patients sometimes. So those microorganisms have the chances of causing infection to us, which will take a long time to be cured. So that is biosafety level 3. And then when you work with like AIDS virus and all, so that you, or COVID, you work on biosafety level 4. So these are the different precautions we take while working with microorganisms. Always there is gloves. We always like wipe our hands with alcohol. There is mask. We wear mask whenever possible, like so that if we are working with any aerosols, we might take it inside our body. So that is one thing. So next is like it is very difficult. I know it is very difficult to educate a layman when we tell them like we work on microorganisms and we are still not infected. But the thing is, you have to educate them. You have to let people know that when you work on microorganisms, you are working with utmost care, because life is very precious. You are taking good care of yourself and then you are trying to handle the microbes. And obviously, when you do research on microorganisms, you can also tell people that, while we take too much antibiotic, so those antibiotics are going and making the organisms inside us resistant. So when those organisms increase in number and cause disease, we are not able to cure that. because this is because of the overuse of antibiotics. So yes, there is a misconception.
Yes, okay, thank you so much, Priyanka. That has been a very wonderful answer. I think that's much clear to Cosmos and to every one of us. And I thank you, Priyanka, for taking time to answer those questions. Yes, I don't seem to see any other more question from the charts or from anyone else. So I think we're going to stop our session at this juncture. So I just would like to appreciate everyone who made to attend this session today. Our last session of the webinar series program will be next Saturday. I will announce that. And yes, as we keep on also researching on antimicrobials or also maybe investigating the approaches like maybe the current technologies in AI, bioinformatics, and all that to see that we can also contribute to the fight against AMR. Thank you so much, Priyanka, for taking that time. Thank you for honoring the invitation, and we would like to invite you in the future sessions. And
Yeah,
we hope
sure.
that you'll be available for that.
Yeah, definitely. Because I really loved interacting with all of you. And it was really a good session with lots of questions coming up. So yeah, I really enjoyed.
Okay, thank you so much, Priyanka, and thank you everyone. So up to this juncture, I don't think we have more to say. Is just to sign up for session today, and see you next time. Thank you so much.